So, this time I'm going to try and do biology in 10 minutes. The whole of that.
I'll give you a bit more than 10 minutes. I think you'll need more than 10.
Well, 10 minutes to do that and then to talk about it after. So, I think if you're making the case for creation rather than evolution, I think there's two things which you can address. Abiogenesis, which is life out of nothing. You've got some rocks, you've got some water, and then you've got to make life out of it, and it's probably the biggest step of difference between that basic chemistry and life itself. What I want to try and do, and we'll maybe do it a wee bit in the second half, is carry on getting you into thinking this through yourself. All I'm doing is giving you some information that's only slightly useful. If I can manage to get you into thinking this through yourselves more, then answering other people's questions, thinking about it, understanding it, I think becomes a lot deeper. So, what I want you to do is imagine if you were sending some robots to Mars to colonize the planet ahead of time, and you're going to come back later and they'll have survived and they'll have colonized the planet. So you can send a few robots to start off with. What are the sort of things that they would need to be able to do themselves to survive and to fill the planet and make it habitable for themselves?
Maintenance and repairs.
Maintenance and repairs, yeah.
Energy.
Reproduction.
Energy, reproduction.
How do robots reproduce though?
Well,
Build themselves, yeah.
build
They'd
themselves.
have to mine some ingredients out of the
Yeah, so they've got to get ingredients, materials,
materials.
energy, reproduce, and repair themselves. Brilliant. So the simplest life form has to do those things. They've got to gain energy, gain materials, you've managed it all. They probably need to move around the planet, they need to grow, and they need to replicate. Otherwise, if you send two robots, you're going to end up with two robots. But if you want to, and those are all the sort of characteristics of life.
You're going to end up with two rusty robots.
Two rusty, broken
Scraps
robots.
of metal.
Yeah.
Left
So,
here.
if you pare what life must be down to the absolute minimum, it still has to have all those things. You can't have some of those things. It can replicate, but not gain energy. It's still going to die out. Okay, you've got to have all of those things and all of them all at once. You can't have some of them. Now, this is your DNA. Deoxyribonucleic acid. It's a genetic code. Each of these teeth is an encoded letter. There's 4 characters, A, G, C, and T. And to replicate, this is what it does. So this is it has to uncoil this double helix. One coil goes off here and gets really easily replicated. There's something here which helps it do so. This other strand, it needs to come from the other direction. So what it needs to do is it creates a loop, it copies it from the other direction, and then it makes another loop.
Simple.
So, and all those things in the middle that you saw, and then it coils itself up. All those things that you saw in the middle are proteins, protein machinery, which have to be made and are encoded in the DNA strand. So the instructions for making those proteins are in the DNA which it's copying. This is how it sort of is within within the cell. So you're in the middle of the cell.
This is every cell.
Every single cell has 3 billion characters of DNA, which give the instructions, and it's arranged in chromosomes, which is like if you take the chromosomes as books full of letters, and the DNA is letters. To segregate, what it needs to do is it makes those copies, which you just saw, and then it pulls them apart by some mechanism, and it makes 2 cells. That's the basis of you going, as you were created, as one cell with half your mother's DNA, half your father's DNA, one cell with enough information. You've just made 2 copies, and you've got, this is how it pulls those 2 copies apart. One to make one new cell, and one to make the other new cell. You've got 23 books, 23 chromosomes, and what it does is it makes these microtubules, and it grows these little thin fibers, and it grows them until and it has to send signals. If you think about how this all works, somehow the cell which became a skin cell on the end of my finger needed to know where it was in comparison to everything else. So it didn't become another heart cell, didn't become another muscle cell, it became a skin cell. And there's all sorts of sensing. That's what things like your hormones are and stuff like that. It's sending out signals, and the cells are doing it. These are what's going on inside the cell. That's kinesins and things like that, sending information along these tubules. And once it's finished using it, those will get destroyed. Otherwise, your cell will end up getting too solid. So that's the simple mechanism that you go from one set of DNA, 3 billion characters in human beings, less than that, but your minimum amount of information is still a lot. And that's what you need to happen to copy one bit of DNA, uncoil it. It's basically a ladder. And the way it works is that if you've got it's like a photograph and it's negative. You get 2 copies, one's the opposite of the other. And if you split them, then you add another negative to the positive, because it knows how to copy. And you need another positive for the negative, and you end up with 2 copies. But that's what it takes to do it. I'm going to go back right to the beginning. Hang on, I can go back here. Hang on. Let's go back. I need to do that, and then that. So that's the code that it's copying, and that's the code which makes you, with some of your father's characteristics, some of your mother's characteristics. If that code was different, and it was a code like in an elephant, what you would create from one cell is an elephant. These bits, that blue bit there, and that purple bit there, and that green bit there, are protein mechanisms. They're made from a string of amino acids, which then folds together and to make something which does this. And they're catalysts which help that chemical reaction happen. And they're motors that spin at the speed of a jet engine to uncoil that DNA. They're quite sophisticated. And evolutionists will argue that abiogenesis is not part of evolution. But to get life without a designer, without a creator, you've got to have done that. And that's one part of it. We've not talked about gathering energy, gathering materials, growing, all those other things. We've just talked about the replication. And that's a glimpse. It's more complicated than that. It's a little bit like me showing you.
So that's one stage out of the 5
Yes.
you've looked at.
It's like me showing you how a car works and how the 4 stages of combustion in the engine and the drive shaft and the wheels work. I've not shown you how the fuel pump works, the carburetor works, the cooling, the differential. We have not shown you on all of those. That's a simplified form of that. There's a lot of other things which need to happen as well. So I kind of want you to get the picture. This is how stuff is moved around the inside the cell. You have these motor proteins. And what's happening here, these are all made out of atoms of oxygen, carbon, hydrogen, and there's an energy molecule, ATP, which a molecule uses, and as it uses one of those, it will make something move, and therefore it ends up moving along that way, but it's in a coordinated way. Let me just show you. So there's a lot of coiling and knotting which kind of almost goes on. It's what you call a supercoil. There's a coil of, there's a twist on the DNA, but basically it's just a sort of ladder of information. It goes A, G, C, T, 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 G, 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 A, C, D, etc. This little chap helps sort that out when it gets a wee bit, because you're coiling it and it will get itself into a knot. So it has, something like this is made from the code of the DNA. And it's got several parts. It's upper gate, middle gate, lower gate. And 2 loops of the DNA will go into that. This middle gate will will split that, some of the chemical bonds apart, so that this other strip of DNA can slip through, and then it'll put them back together again, and it'll release this lower one. I think I've trimmed it in the wrong place. So it'll release the lower one, so where you've got a loop of DNA which can't, it's just on it, which can't let it go, which can't cross, it will free it.
It'll
And
cut
so-
it and rejoin it.
Yeah.
Yeah.
So these need to know where to go. They need to attach to the right things, which need unlooping, and they need to go through those processes. And it uses the ATP in the liquids to do that, but it's doing a kind of intelligent thing. It's a small mechanism,
Yeah.
and the code to make one of these machines with all these different parts is encoded in the DNA. So you've got a chicken and egg problem, that how do you replicate that DNA and stop it tangling if you need one of these, and the way you make one of these is you need the DNA replicated. And to replicate it, you need all that mechanism at the beginning to copy it and split it off, etc. There's all sorts of other error correcting things which will go down and see where something else has gone wrong in the copying mechanism, and it will correct those out. And if something's very wrong, it will actually tell that, or even the whole cell, to die. And if the mechanism's gone wrong, so there's all those kind of mechanisms built in. If you don't have them, you need that repair mechanism. I kind of want you to get, I think if you get the idea of quite, I hate the use of the term it's complicated, because a pile of sand is complicated. But it's not sophisticated and it's not specific. This is sophisticated, designed and specific. And if you think that can have created itself out of nothing, then I don't think you've understood that. And I kind of go, I don't need to argue this, do I?
This is the interesting thing is this is the last sort of 15 years that really DNA is starting to be understood, and these are it's very recent. And when Darwin was proposing the idea of evolution and science brought into it, it was
This wasn't known.
it
at all.
was it considered that the cell was simple. They didn't know what was going on inside a cell. Now they've discovered all this going on inside a cell. They're going, wow. So there's loads of questions from scientists about that, as there is in lots of other areas, like space at the minute, because of the James Webb Telescope. They're seeing stuff that's completely blowing all their astrophysics out of the water. They're struggling with it because they're seeing. They should be seeing young universe, but they're seeing fully great universe. So it doesn't look like it happened the way they think it happened. So it's good stuff. Okay.
So, yes, as you say, I mean, Darwin thought it was simple. And there's a guy who's actually a organic chemist, a sort of guy who has won the competitions of getting a nano car, which is made out of specifically building molecules which have axles and four wheels and a motor in the middle, and if you shine the light on it, the motor moves, and they get it to go over a grid of or a sheet of gold or silver. They tend to use a guy called Dr. James Tour, and he's made comment on this and sort of said, the chemistry just doesn't work. You can't make it do this without a very specific
Program.
environment and program, and you need the cell to make that happen. But what you're trying to build is the cell in the first place. It's another chicken and egg problem. And one of the things he's saying, as David was saying, is that the origins of life research is getting further away from the target. Because although they're saying, we can do this chemical reaction, we can build that little bit, and we can do this chemical reaction, we can do that little bit, at the same time they're learning more and more about these complicated interactions of the chemistry and the systems and the mechanics of it, and the target is just getting further away. So they're further away from creating a cell than they were 20 years ago, 50 years ago, even 5 years ago, whatever. The target's getting further away because we're learning more. So that's what's, so we've kind of seen what's in the nucleus of the cell. The whole cell has got other bits which are creating stuff, moving stuff around. There's a lot more to it. I won't labour that point. Just to give you an idea, so to go from the information on the DNA to making one of those machines, if you remember the one which sort of unknots the bit, and it's got a number of different proteins, what it will do is every 3 letters it will make it will pick up an amino acid. So that's the code that it uses. in computing, we use a thing called ASCII code. So that there's an with the original ASCII, there was an 8-digit binary, which a certain pattern of 10101 was a capital A, and a slightly different one was a lowercase c, etc. This, the DNA is a code which tells you what amino acid to bring up. And you've got amino acids which, if you put a few of them together, will pivot, other ones which are rigid, some which will create an electrical signal when combined. So it's like Lego blocks which you're putting together. It then needs to go and be folded up, and they're struggling to work out how that happens. And you make out of this string, normally thousands of characters long, that folds up into a mechanism that does something. So I'll give you one instance. On the hairs of your ear, your hearing at the moment, you're hearing me talk. That's just pure pressure waves through the air coming into your ear. Your ear is vibrating. That's translating mechanically into a fluid in your inner ear, which is vibrating. And there's a spiral in there, and there's hairs of different lengths. And each of those hairs of different length vibrate at a different frequency. And it's called the fast Fourier transform. It's each of the hairs vibrates to a different frequency, and that's how you're analysing what I'm saying to you. And electrical signals are going from there into your brain, and you're interpreting that as meaning. The engineering of it is just insane. You're interpreting that as meaning. Now, the way...
It's not making any sense, James. You are. You're doing a great job.
The way that you get that movement of the hair into electrical signal is the DNA will pick up, will get translated into a string of amino acids that will fold into a protein called prestin, which will coat the hair of the hairs in the inner ear. It only produces that when it's in the right place and it's going on the right area, and it produces that and it coats that. And as you strain that, as that hair moves, it creates an electrical potential, an electrical signal. And that's what's sent to your brain. Okay, so that's just one little bit which is going on inside your ear to make you hear what I'm saying. Things like muscles have kind of gears, and as you give them an electrical signal, you give them pulses of electrical signal, it will push, switch that one off. It will push, pull one fibre against another, and it will contract. So our bodies are full of these mechanical things which is hard not to see as designed. So I'm kind of going on really into how evolution would work. To create a new design, you need to create new proteins. It's a little bit like kind of a protein is like a bolt or a nut or whatever. You then are going to create your car out of these components. You've still got to put them together, but you've got to start with these things which do stuff. And bolts are only useful if they're round. They're not useful if they're triangular, square in cross-section. They've got to be round. You've got to make them right. And most things that you try and make will make rubbish. It's like typing. If you randomly type, most of what you type will be rubbish. A very, very small percent will be useful. And you've got to make this into something which is useful. So to make one protein is thousands of characters long in the DNA code, translated by a language which is embedded in all human life. And the way that they claim evolution works is that there'll be a copy error along this string, and that will change the protein or even create a new protein. You need to, you've got to start with one protein, and then the organism creates more proteins, and then creates more proteins. There's more proteins created in a human being than there is in an onion. So you've got to create new proteins, and the way they say that you're doing it is by copying errors in this code, which will
Happen
create
to be good.
happen to be good.
Beneficial copying errors. Yeah.
Now there's 22 different proteins, amino acids, and 48 different codes which can be programmed. When it's copying that along, it's copying a few thousand characters, normally tens of thousands of characters of these amino acids which it's putting together. One of those codes is a stop, what they call a stop codon. If it has a particular, I can't remember what it is. But let's say,
STOP maybe?
say it's AUG, and if you have a mutation and you suddenly create that, it will just stop. So if you just by mistake, 1 in 48 chance, have a mutation which creates a stop codon, you're going to be copying that protein, and it's going to stop. Most of mutations will create an error. And I'll just show you. So, these are different sort of errors that you can get, and it's a copying error. And if you imagine a text which sort of says create an arm, create another one on the other side, etc., you can understand how mutations work. That or don't work. What it's doing it's just creating error inside that, and the chances of you changing one letter or a few letters and getting a meaningful word and getting a meaningful sentence and a meaningful paragraph in a chapter which makes something useful is pretty slim. So you can delete it, you can duplicate it, you can have a bit going upside down. You can translocate it and put it into the wrong place and stuff like that. All sorts of different types of mutations that you have. But basically, most of them are SNPs, a single nucleotide polymorphisms, a change of a single nucleotide. This here is number 10 on the your chromosomes, the 10th book in your library. We've got 23 plus the X and Y chromosome on the right hand side. That's the 23 listed out there. They list them by number 1's the longest book. They do it in a really simple way. So now this is the mutations that we know of on chromosome 10, and these are the diseases. You won't be able to see them from there, but these are listed here. So opsonic defect, chronic infections, retinal non-attachment, and neuropathy, half of them I can't even, Graves' disease. Each one of these is a disease caused by a mutation in that location. And we know that, and we can test for that now because we can read someone's chromosome. And you'll see that sheet over there, the thousands and thousands of diseases associated with mutations. The idea of evolution is that a mutation will be creative. It will create new information that will turn a dinosaur into a bird. It will turn a fish into a frog or something like that. It will create some new information. It will create feathers rather than hair, which is, if you look at the design of that, is very intricate, very complicated. It's going to take a lot to do that. And the argument is that you can take mutations and that will be creative. What we observe is that mutations are destructive. Those are all the diseases that mutations will create. If you asked what positive things we have observed which mutations have created, there are about 4 that I know of, which they will argue. I could list all of them, but I'll give you just one, which is sickle cell anemia protects you against malaria. Sickle cell anemia means that instead of your blood cells becoming like a nice healthy donut, it's the wrong shape, and it's a sort of it's like a sickle, which is why it's called that. But instead of that blood cell living in your blood for around about 60 days, that's what your blood cell normally does, and then it will die. It will only live for about 20 days. And because it only lives for about 20 days, the effect of malaria doesn't have enough time to develop. So people who have sickle cell anemia won't get malaria. And you go, well, that's great. So the prevalence of malaria has increased in areas, the prevalence of sickle cell anemia has increased in areas where there is malaria, because the people who have sickle cell anemia benefit. So that's a beneficial mutation. I think hopefully you can see that although that's one of the best examples of a beneficial mutation, that you're not increasing information on the DNA. You're not making it better. I'll give you another example. I like to use cars because.
What are the negatives of
Sickle cell?
Well,
I was thinking that because I'm pretty sure I've heard that. It's not a great thing to have.
It's not a great thing to have.
What
No.
push on your kid?
I mean,
You'd rather just treat the
because,
malaria when it comes along.
because your blood cells are not the right shape, they're not absorbing enough oxygen, your body is devoid of oxygen. I mean, low energy is the least of your worries. It's really not a good thing. There are things, and some of the examples of look at bacterial resistance to antibiotics and things like that, and you can look at those. That there are. So there are examples which evolutionists can bring and say, but look, this is something which, through mutations, has aided something. And I'll give you an example in the car world. So if I wanted to get from here back to Selkirk quickly, I could rip off the windscreen wipers, rip out the back seats. I could make the whole vehicle lighter, and it would be better at that one thing.
Going fast.
But
You're
I've done it.
What if it rains?
I've done it by destroying things.
What if it rains and you can't see?
The resistance, antibiotic resistance, often works because the antibiotic attacks a particular part of the bacteria. So imagine something people are dying of athlete's foot people without feet are going to survive better. And that's the way it basically works. Not in every situation, but in most situations. So if a antibiotic attacks a bacteria in a particular way, and a mutation happens in that, a lot of things happen with these sort of key and code. They hit each other. Sorry, I'm going to go back. And if that a bit of that key, that code lock changes, then it's not going to happen. So that's how a lot of these things work. So COVID worked in an absolutely classic way. It's the same way as Spanish flu. You can draw the graph, it rises, and then mutations start happening. Human beings work out how their immune system works out how to defeat it, and it tails off. And you can look at those graphs. Sometimes you get a second wave, and then there's often we had the alpha, beta, gamma, delta. I had the alpha and followed by the delta, and they're variations, mutations, and they attack you in a slightly different way, and your body isn't used to that slightly different thing, and it comes off. But you notice that, although as we now know it came from a lab and it was genetically engineered, it is degraded, and
until it fizzles out.
Until it fizzles out, and that's the normal way of these things. You get something which does attack us. And that may be a weakness in us, but that's anyway. I don't want to get into that too much. Okay, so I want to give you an idea of how to look at, because you know if you should be kind of going, yeah, but how about Darwin's finches? How about peppered moths? All these icons of evolution that we've seen, where the moths in Britain were white
colour, yeah.
and dark ones, and then in the Industrial Revolution, when all the trees and everything was covered in black, then the dark moths survived better, and that was surely that was evolution. We saw the variation of the population change from white moths to dark moths, and then after the Industrial Revolution, when we stopped burning so much coal, we went back to white moths. So how do we understand that? I would say that there's 4 mechanisms that, is it 4 or is it 3? Anyway, four mechanisms which happen. There's more DNA than we have. Our parents had 2 strands of DNA. My mother had 2 strands of DNA. My father had 2 strands of DNA. So that's 4 strands of DNA. I've only got 2. So I've only got half of the information from my mother and half of the information from my father. But I've not got the same mix as my brother has. So I look different, unless I'm a twin, in which case it created one cell, and then it divided and then became 2 people. Identical twins, sorry.
I'm a twin. Don't
Yes,
look
yes.
at that.
So you've got a range of information there, and you're only selecting some of it. So I would refer to that as variation. I'll give you an example. When they designed Woody in Toy Story as CGI, they designed him as one person. When they designed the orcs for Lord of the Rings, they didn't design every single orc. What they did was they designed a generic orc, which could be anything from this height to this height, anything from this width to that width, or this ratio was this number to that number of scars, etc. They gave a range, and they did a very similar algorithm as we do with humans, and that's what we have. We have this sort of variation. So there's there's more genetic code, but that's and if you think about it all in terms of information, the information is already there. I'm selecting from the information that's already there. I'm not creating any more, and yet we have the variety which is in this room. And you can take two identical people. You can take two identical dogs or two identical wolves, and you can breed them, and you will get a wide variety. You will create new varieties that you don't have. You'll go down some cul-de-sacs, etc. But there's a load of information, and you're selecting, and you're out of that. And the way it's done is very good for doing that. So you can see that. The reason why Darwin came up with this idea, he was very much at the time when people were breeding horses to be faster, dogs to do different things. How can we get the best apple tree? And they worked out what a genetic of a tree which produced a brilliant root structure, and they grafted every apple tree that we have now pretty much in the world onto that. And different breeds of apple tree which go onto that. He's looking at it and going, well, if we can do that with dogs and horses, and we can make horses faster and faster and faster, then maybe naturally selecting things, we can create new things. And you take, yes?
That comes back to where you started a couple of weeks ago. We can create variation, but we can't create a new species.
Yeah. So it's within, one of the easy ways to look at this, it's not often looked at now, is the number of chromosomes. We have 23, chimpanzees I think have 22, is that right? Or is it, they've got 24, other things have 32, etc. You kind of go, how many books have you got in the library? How did that happen? There is some arguments about, well, those two combined and that made this one, etc. And you can pick those apart. But anyway, you've got this variation, no new information. You've got adaptation. If I take my socks and shoes off over summer and walk around on gravel, the skin of my feet will get thicker. If I go to the gym, not likely, but and lift weights, my muscles will adapt. They will realise that they're being stressed, and they will build up. Again, I'm not creating any new information, but I am changing. If I go out in the sun for too long, particularly as a white guy, my body will say, I need to protect myself from too much UV radiation. I will tan, and that will protect me. So you're not that mechanism is built in. Although I have changed, my DNA hasn't changed. With variation, although the group of people have changed, the DNA hasn't changed. The peppered moths, you had white peppered moths, you had black peppered moths. If you kill off most of the white peppered moths, then you will have mostly black pepper moths. But you're still creating both of them. If you killed off, if you killed off all of the black ones, you would only ever have white ones from now on.
Wrong. Surely.
It can be.
Yes. That way round,
because you've got the information still in the white one to make a dark one.
Necessarily.
Actually,
Yeah, not.
let's take it to
Depends on the species.
Yeah, let's take it to hair colour, because we kind of see that quite easily. There's two colours of hair. There's black hair and there's brown hair or red hair. And if you're grey, I don't mean gone grey, but if you're grey, that's a partial expression of the black. If you've got both, then you're brunette. If you've but a redhead marrying a redhead, if you're a pure redhead, then you've got none of the black available. The black will cover the redhead, so it is more prominent in hair colour.
More dominant.
It's more dominant. So, but in say the Chinese population, there's predominantly black hair. But you can end up losing something in particular.
Yeah.
And someone with blonde hair is not producing either. Now, often it is still recessive, but if you go down one track for too long, and the reason why you the Bible, later on in the Bible, not right at Genesis 1, they intermarried, but later on it says you can't marry your sister and your cousin, etc., because you've got two strands of DNA. If on one strand and that's one of the ones here, you don't produce insulin, it's okay, you've got another strand.
Yeah.
You've got two copies of the insulin of insulin. If you start intermarrying with people who are the same as you, your chances of having both strands where you can't produce insulin is and you get to the point where you can't go back, because all that group of people haven't inherited the means to create insulin. All human beings have the code to create vitamin C. It's broken in every single human being. Dogs have it. Dogs do not need to eat oranges or lemons or anything else. Human beings do, because we cannot create our own vitamin C. It is broken in all of us. I suspect Noah had that broken gene, and so did his family, and it's what's called, it's fixed in a population.
Mm-hmm.
Crufts.
So some things are fixed.
Crufts have problems, and they've gone away from this absolute pure pedigree because it becomes inbreeding, and you have real problems. Manx cats don't have tails, but 90% of them are in these cat hospices because they are incredibly ill. They are inbred and they have the same mutations. So you can have lots of variations of mutations, but if you have the same mutation on both strands, you're stuffed. You can't do anything about it. Each generation you will have, I have about 100 to 200 mutations which my parents don't have. I think that blows the whole sort of evolutionary idea of mutation and selection out of the water. If you had one mutation per 10 generations, and you could then select from that, you go, well, that's a good mutation, that's helpful, we'll keep that. But even if I produce something which is randomly kind of doing something helpful, and therefore I'm slightly better for that, I've also got about 100 more mutations which are harmful. And we are accumulating mutations. I just realised there was something else I was going to do, and I promised I was going to do. I will try and find it for the second half. So we've got different sorts of DNA. I'll go back to the cell for a moment. I've been going on a bit too long. Most of our DNA is like, if you view it as the design instructions, most of it's in the office in the middle, in the nucleus. But some of it is the DNA for making what you make lots of. So just like a factory, and viewing this as a factory is a very apt description. If you go into a factory, you'll find roundabout design information for making the screws that you use everywhere. Because you're always making those, you're just always making them. So you need lots of them, so you have the instructions locally. And in the mitochondria, where a lot of the stuff is made, there's a small ring of DNA called mitochondrial DNA, and it's about, oh, I should be able to work this out. Hang on, let me step back. There's guys have, men have an X chromosome and a Y chromosome. Women have 2 X chromosomes. The Y chromosome is only passed down through the male line. So unlike the sort of everything mixing around with variation, the Y chromosome is only passed down through the male line. So you can track that, and if there's a mutation that is in the Y chromosome, your children will have it. You haven't got another option. Your children will have it. And that is accumulated over time. And
so your
that,
male children will have it?
Yes, your male children will have it. And that's reasonably big, so you'll have about 3. So that's about 3% the size of the whole chromosome. Your Y chromosome is about 3% the size. The mitochondrial DNA is only in the cell. So you only get that from your mother, because the sperm doesn't have any mitochondrial, it doesn't have any mitochondrial DNA in it, so you're getting no mitochondrial DNA information from your father. So you can track the female line through the mitochondrial DNA. And the mitochondrial DNA is really small. That's your common little nuts and bolts information. To be honest, we don't know what most of this stuff does. And anyone who tells you they do is lying to you. We know that all of it's used, and what diseases is caused with bits that break, and that tells us as much as we pretty much know. So the mitochondrial is about 1/10 the size of the Y chromosome. So about every 3rd generation, you get another mutation on that. Now, we should know, if we look at DNA from historic graves, and from comparing humans around the world, particularly if you look at something like mitochondrial DNA, you should be able to know how many generations back we've gone. Because we know that there'll be this tree of this mutation happened in this family line, and all that woman's descendants have that mutation in it. And you can track that, and one of the things which is happening around a very good book called Traced, which you can tell, you know, which way did the Polynesians get to their island? Did they come from the African continent or the Americas? And you can track that, and you can see which way they went, and you can look through. So we can, and you can tell how varied that's become. And what I'll do is I'll look up the image, because that actually also tells us how many generations we've had going. And of course evolution has predicted how many generations we've been going for, and creationists have predicted how many generations we've been going for. So tracking that mitochondrial DNA is another thing that we can do. I think I need to shut up. Any questions, sir? I get to the end of that?
So one of the key things is with Darwin evolution, we've got the tree of life, and we start with the goo to you, so you know, lightning hits a puddle, and all this marvellous stuff happens. We produce a first replicating cell, although that's totally ridiculous, because to get the first cell you need all this stuff, and even, you know, by lightning a thing we can create in the lab we can create amino acids and the like, and we can probably create some proteins, but you have to create them all together. The proteins start breaking down, within a couple of days. There's nothing left of them. They're not functioning. They're breaking down, because that's what happens with life. A lot of these biological components, they're breaking down. So for this puddle to create all these wonderful things and get them all together at the same time, within a cell wall, and get them functioning and replicating and whatnot, it's just pie in the sky. It really is. And we would say, biblical creationists would say, we're talking about an orchard of life, okay? That God created all these different kinds: humans, chimpanzees, frogs, dogs, and that they all have a tree that they've become different things. There's lots of different sorts of humans, okay? We've got Asians, we've got Indians, that look differently, and we can categorise them. We've got different sorts of dogs in that orchard, that one tree. They've got all that species, but there is not any movement between the trees. We have an orchard of life rather than a tree of life. So anyway, these are, and it speaks into the somewhat. Yeah.
So I've just picked up those pictures I was looking for. This is from Is Genesis History, and if you go with the biblical model, your DNA is very much affected by a couple of events. Noah's flood, you suddenly went from millions of people to 3 family lines. Noah with 3 sons and 3 daughters. So the 3 daughters would have daughters-in-law, 3 wives would have presumably had different DNA, and particularly different mitochondrial DNA. So if you look at the middle of this, you would have a certain amount of mutation going on for a while, and you'd have those family lineage. You're basically drawing the family lineage. So you've got 3 daughters which are surviving. Anything else which spurred off somewhere else, you're not going to have. You've lost that in the flood, unless you find some. If you dig up anyone who was killed in the flood, and then from there you'd have different family lines spreading out, and because we know the mutation rate of roughly 1 mutation in the mitochondrial DNA per 3 generations, you only actually have something like between 32 and 36 predicted mutations in each line. So that's what that model looks like. If your model is millions of years, it will be the same as that, without the 3 in the middle, and it'll be millions of years, so it'll be thousands of mutations, tens of thousands of mutations. And that circle will be vastly bigger. So the idea that creation is not science because it's not falsifiable and it's not predicting anything, it's nonsense. We can look at the Bible and say that's what it says happened. That is exactly what you would predict you would see in the mitochondrial DNA. These are in different directions just because they're different mutations and different lines. And if you're along this line, you and your daughter and your granddaughter will have the same mutation along that line.
So what are you saying? You're saying that the mutation, the errors that we've got in our thing suggest that we've not had that many generations.
Well, that's a prediction. That's just a prediction.
But that's what you see also, is it?
Okay, so if we actually do the test, on the right, that's what we get. That's the reality on the right, that's the theoretical prediction on the left.
So it's not that far away.
Well,
If it was,
who knows? I don't know.
if you were talking about millions of years
Yeah.
of human life,
Yeah.
it would be hugely bigger than that. It goes bigger. Okay? And what we have, it is more complicated, but what we do have is we have these three nodes in the middle. And not all of the African ones come out of this node. You can have a look at this on the screen later if you want. This one, which goes there, is the Europeans and some of the Asians, and some of the Asians are here as well. The three sons of Noah, who can tell me who they were?
Shem, Ham, Japheth.
Yeah. And where did Ham go?
Africa.
Yeah, Ham went to Africa. So this is how you can actually kind of tell. You can follow the lineage. If you go to Uganda, my brother worked in Uganda.
I was born in Tanzania.
You're not Ham either. I can tell. And they will refer to themselves as Hamites. Shem is the Semitic people. That's where the word comes from. They trace their roots from the Semitic people, so that's where Shem. It comes from that word. And Japheth, and the Bible's got predictions about where they will go, and that Japheth will live in the tents of Shem. That's the Japhethites. Yes, so that's the Japhethites living in the house of Shem. Is that right? Anyway, you can work it out. I can't at the moment. My brain's not doing. But some of those in Africa are Semitic. They come from the Semitic line. This is the Semitic line. And then Japheth is there. So you can even actually track it there. But the size of it, the scale of it, the pattern of it, is almost identical to the prediction of a short Earth. So I have no problem with a longer Earth, a longer geology, a metaphorical interpretation of Genesis, which tells me biblical truths through the story of Noah. But the science clearly says that actually we've only been around 30 odd doublings. What's it work out? 900-ish generations, something of that nature. And that's what is.
So 900 generations, 50-year-old generation would be 4,500 years.
Well, it's for a generation about 25 years.
25 years. Yeah, so it's not because it's
So in the Jewish culture, they track their lineage through the mother's side, isn't it? So
they do now. Yes, that changed with rabbinical Judaism, but yeah.
Okay.
Yeah.
So could they theoretically track their lines all the way back to?
Well, what really amuses me is you had the Levites and the Kohanim being the priests of the tribe of Levi, who were the priestly line. And there's a joke coming because what would be interesting would be to get the Levi genes, which you can trace. I was ready for it. Let's get a coffee.